את זה היום, בסדר? זאת אומרת, אני יודע שזה לא עוזר, אה? לא נורא. אוקיי, טוב, יסלח לי שאלה למבחן כן. האמת שאני כבר מבקש מאלי נרקר לשלוח לי את המצגות, כדי שזה יהיה מוכן שבוע. תודה רבה. אני לא בכדי חובק קצת.
עדי, אם אתה מבקש מאלי את המצגות, אתה יכול לבקש גם את המאמרים בסשן שלו?
כן.
לעשות איזה שלושה שבועות.
כן. טוב, עכשיו. אז להדליק פה את ה... שומעים אותי למיקרופון? לא. צריך להדליק. מה עוד נצטרך להדליק פה? מחשב גם מצלמה, דימר, מקרן. שיודע? אני לא מצליח להדליק את זה. מה עוד צריך להדליק? טוב. English, right? So...
עובד. You're all set.
Just turn off my own cell phone. So, good noon, everybody. My name is Yoram Ben Shaul. I am at the Faculty of Medicine, and I came by to talk to you a little bit about the Vomeronasal system, which is another chemosensory system, just like the main olfactory system is, which Adi spoke to you a lot about. And I think I should also mention the taste system, which I think you will not learn about this year, but it's important to stress that you're only sampling a subset of the chemosensory systems, and I will mention some of those as we go along today, but concentrate on the vomeronasal system. This is an organ that we'll talk about in a few slides, and its role particularly in chemical communication between animals. So between different animals is the stress. So let's start with some definitions that are somewhat dry, but it's good to have a general framework for what we'll talk about. So chemical communication, of course, communication between animals is important, and chemical communication is a very important mode of communication between animals. Some animals more so than humans, but even in humans, I argue that this is a relevant mode of communication. And we talk about semiochemicals, that is, chemicals that can signal information that organisms can extract about each other. And so there are several categories. One of them is actually not really a semiochemical in the sense that it acts within an organism, and these are hormones, but they're probably the precursors of some of the other semiochemicals that we talk about, and they mediate information within the body. I think you probably heard about them. We will not talk about them at all. Then there's another category, which are allelochemicals, which relate to chemicals that are serve to communicate information between different species. And then we can divide it into different categories. Chiromones are those that benefit the receiver, so that means if you're a receiver, you benefit from this. So you could be a predator that you can detect prey, or you could be prey that detects the existence of the predator, and that helps you to get information, to either to survive or to get food. Or it could be that the sender benefits. For example, you can lure another animal to come by, and so you can prey upon it, and then in that case, it's called an allomone. And cinnamones are, sounds like cinnamon, but cinnamones are chemicals that both species benefit from this. So these would be chemicals that serve for symbiosis. Now, the ones that are highlighted, which we'll talk about more, are the pheromones, and these are chemicals that are communicate information within a species. And there are a lot of definitions, as I already said here, and there's a lot of ambiguity and fuzziness, and there's also overlap between pheromones and the allelomones that appear here in different categories, because a given chemical can serve different purposes, and this often happens in the way that these systems evolve. But when we talk about pheromones, the original distinction made the original classification made a distinction between releaser and primer pheromones. So a releaser pheromone is one that elicits an immediate behavioral reaction, whereas a primer pheromone is one that primes a certain, usually endocrinological process or physiological process in the animal that receives it. And these, again, there can be some overlap between these two categories, but that's a distinction that was made, and it was especially clear in insects. So the whole concept of pheromones was originally coined for insects, and that is where it makes most sense, and as does this distinction. Now, another class of chemicals that usually people speak about as pheromones they do not really immediately release any type of behavior or process, but they communicate information about the individual. So these serve for individual recognition. These are usually complex mixtures, or at least a mixture of several components, and they do not fall into these two different categories, not releaser nor primer pheromones. And we'll meet different categories of these as we talk today. So let me, before we get into the vomeronasal system, a few more words more generally about the use of chemicals in animal communication. So animals develop specialized mechanisms for advertising and spreading their information around. You probably know that urine is a very important source of semiochemicals, and here you see a mara, which is a type of a big rodent that actually sprays urine. A male sprays urine on the female, so whether she wants to or not, she has information about this specific male. Here you have an example of an antelope that's depositing scent marks from a special gland on some twigs, and other animals of the species can also detect this information. Usually these would be territorial marks, but not necessarily. This is a very extreme example, not extreme, but a very intriguing example of a male bat that has a special sac that's called a wing sac, and in this wing sac there's a mixture of a lot of stuff that's not very appealing, I think, to humans. It contains the dung, it contains the urine, it contains also secretions from other glands, and all of this is mixed together, and then the male just wafts this, makes the airflow bring these scents to the female, and presumably she gets a lot of information from this type of secretion. And this, of course, is an example from humans to make the point that humans also make use of scents to advertise how attractive or nice or pleasant they are. Yeah. I think female. In this case, it's female. Probably other males would be spreading it in the territory and not directly on other males. I think that would elicit an aggressive reaction, probably, or a submissive one. But in this case, it is a reproductive kind of signal. Okay, now the sources of the chemical signals are also diverse. So there can be, we already met some of those. They can be specialized glands. For example, ants have a lot of different specialized glands, and each type of gland produces a specific type of chemical that communicates a specific type of information. But we're talking mostly about mammals, so here also in reindeer they have 5 major glands that all of them are used for emitting information. And also skin is, of course, a very big organ that can have secretions that, including glands that secrete products that give us information about us or other animals about their conspecifics. Another source would be plant-derived components. So animals don't have to generate, to synthesize whatever is used for chemical communication from scratch, but they can use chemicals. For example, this bee that uses information that's not information, but chemicals that are present in orchids that they use to pollinate. And this can be a sex attractant, it mate attractant. Another example is the byproducts of bacteria. So bacteria, of course, their waste product can generate, the metabolic product can generate can be odorous, and this can be used, for example, it is used in primates to communicate the vaginal or the estrous state of a female, and this is information that is very important for a male of that species. Do you know where humans also have, where some of our own odors are dominated by bacterial secretions? The what? Well, breath, yeah, breath could be to a certain extent, especially when it's unpleasant. You're right, but also the armpits. That's a very we have microflora in our armpits, and that can generate some of the smell that comes from there. So there were three examples, and here is probably the most dominant example, or the most common example, and that is waste products, which we already mentioned. So urine is one such case. Feces is another case. This is deer scat, which are common to see on the trails around here, even in Israel and Jerusalem. There's another type of more exotic secretion, which is castoreum in beavers, which is also some kind of a mix that's probably specifically designed to emit this information. And this is another example that we will also meet. This is more of a specific, this is a specific chemical that's produced in urine, but in this case we're talking about conjugated steroids. So steroids are molecules that play a very important role in the physiology, and they also have their presence says important things about the physiology, but they're not soluble in water normally, so they have to be conjugated, for example, to sulfate. So when they are, they can be excreted in urine, and we will meet some of these chemicals in some of the upcoming slides. So I think these are the examples that I wanted to show you about the sources and the chemicals that are used for communication. And now we can start talking about the system that is used to detect this information and to make something with it. And in insects, there is a very clean, or it seems at least clean, distinction between the main system or a main olfactory system and one that is specialized for detecting pheromones. So there are distinct receptors and distinct pathways in the nervous system for these two types of behaviors. And there seems to be something similar in mammals or in vertebrates, not just in mammals. And this distinction is between the main olfactory system, which is indicated here, and the vomeronasal system, which is the subject of our talk today. But the distinction is not, I should already say up front, that it's not that clear, or it's not that dichotomic, because the main olfactory system certainly plays a role in social interactions, and it's not clear that the vomeronasal system is exclusively related to these things. For example, we do know that the vomeronasal system also processes information that appears between different species. So if we follow the strict definition of a pheromone, it's not just a pheromone system. And so I'm not going to go into the details right here, but I will do this either in the next slide or in one or in a couple of slides. But I should mention that they're not just two systems, there are actually more, and it depends how fine we want to be with our classification. So in fact, even in the main olfactory system that Adit told you about already, people make distinction between various subsystems depending on the different types of receptors or the receptor cells that express them. I'm not sure if you talked about them, but different receptors or different cells might have different stimuli that activate them, and they might also project to different regions within the olfactory bulb. So that's one fine point. Another system is the accessory olfactory system or the vomeronasal system. So these two terms, not to confuse, are used interchangeably. We prefer the vomeronasal system because the accessory olfactory system makes it seem like it's less important, and I don't like to think about it this way. And another, the superior olfactory system, yes. And we also have the septal organ, which is a collection of sensory cells in the nasal epithelium that also has a special projection site. We have the Grunewald ganglion, which is another sensory organ that recently given some more information. This system is probably related to detecting alarm signals, at least in mice. So it's a very interesting system that's probably dedicated. The septal organ, it's not entirely clear, but it may be some general detector of odorants, and it also has sensitivity to mechanical flow over it. So we have several such systems. I just wanted to mention this. We also have the trigeminal system, which can detect painful stimuli or nauseous stimuli in the nasal and oral cavity, which is also can detect chemicals. Yeah.
What's that mean, mechanical flow?
Just the airflow. When you breathe, the air is flowing over the sensory neurons.
What would be the function of that?
Well, what would be the function? It's not necessarily known, but one of them is to enhance the response. Maybe the system can detect the actual breathing and can enhance the response, the sensory response, by some other downstream pathways. Um, and and it could also maybe synchronize the activity in these neurons to the flow of air. But these are the um, things that I can think of. So now about the vomeronasal system specifically. So we leave all the other sensory systems and the subdivisions. We talk about this system, and the best way to explain at least the initial components of the system, I think, is by comparing it to the main olfactory system. So you see here a cut through the nasal cavity of a mouse or a rodent, and this is the main olfactory system on top. So we have the nasal epithelium, we have sensory cells that project to the olfactory bulb, you know that, into glomeruli in the olfactory bulb. And what is the way by which odorants enter the nasal cavity? We mentioned this, and you know this, of course, even before you took the class. Just by sniffing, just by breathing, the odorants enter the nasal epithelium, and then they activate the sensory neurons, which relay the information to the main olfactory bulb, which is shown here in green. So it is main in the sense that it's big and bigger than the accessory olfactory bulb. So at the bottom, you see the vomeronasal system. And there are similarities, but some important differences. So one of the similarities is also that we have sensory neurons. And they're also located in the nasal cavity, but in a specialized organ that's called the VNO, the vomeronasal organ, which I will talk about in some detail. And here you see these sensory neurons, and they also project their axons to the olfactory bulb, but not to the main olfactory bulb, but rather to a specialized region within the olfactory bulb that's called, as I mentioned, actually, the accessory olfactory bulb here. And one of the distinguishing features between these two systems is the mode of stimulus entry. So here we said it's breathing. Here, breathing is not sufficient usually for stimuli to activate the neurons. They have to actively enter the vomeronasal organ. And this is possible only for stimuli that can be dissolved in the fluids in the nasal cavity. And once I explain how the system works, this will probably make more sense. So I think this is all I wanted to say, except also the point that these two systems have very different projections into within the brain, within the central, or downstream in the central, upstream in the central nervous system. And this was already shown in one of the previous slides, but it's made more explicit here. And I think I put this slide right here. I know I put the slide right here, which is supposed to show the flow of information in the system. So we have stimulus, we have the VNO, we have the accessory olfactory bulb, and then we have two structures that are more specific for this system, than to the main olfactory system, which is the amygdala and the hypothalamus. And as you probably know, the hypothalamus is a very complex brain region that contains many subpopulations of neurons, but they can control directly endocrine processes, so the hormonal status of an animal through its projections to the pituitary, and they can also control behavioral outputs by acting on neurons in the brainstem. So this pathway makes it very logical to think that the system has a role in eliciting behavioral or physiological processes, and that's one of the findings that put the study of the vomeronasal system in the context of chemical communication. Yeah.
How many types of receptors are there in the VNO?
So we'll talk about this, okay? I should proceed. Okay, and so this, I said that this makes the system likely to serve a function in physiological processes, and one of the most famous processes, which was actually the thing the phenomena that got me interested in this system in the first place, is the Bruce effect. So have you heard about the Bruce effect? Have you heard about it from me? No? Okay, so did everybody hear about this already? No. So let me tell you a little bit quickly about the Bruce effect. The Bruce effect is the following effect. So we have a female mouse, and she's impregnated by the. So we have a female mouse. She's impregnated by the male brown mouse, and she has brown male pups. And then, if she meets or if she's exposed to a male that she has not met before, that's a strange male, in a very high percentage of cases, there will be a termination of the pregnancy. So she will not carry to term. And one of the nice things about this, what makes this really appealing, is that if she's exposed again to the same male with which she mated, there will not be a termination of the pregnancy. So sometime during the course of intercourse, she forms a memory to this specific male,